Hi, my name is James Hartnett, and for my project, I analyzed three genes found in Myothermus ruber, otherwise known as M. ruber, and compared them to three genes found in Escherichia coli, otherwise known as E. coli, which are involved in the streptomycin biosynthesis pathway. The locus tags of the genes analyzed in M. ruber include MRUB2052, MRUB0628, and MRUB2034. The locus tags of the genes analyzed in E. coli include B0688, B2039, and B3789. The goal of this project was to determine if the M. ruber genes are orthologous to the E. coli genes through the use of numerous bioinformatics tools. In order to begin to understand my project, you first must understand why I chose to study Myothermus ruber in the first place. So it is known that E. coli has been used as a model organism for a number of different research projects because it is so well known and so well studied. But little research has been conducted on other bacterial organisms. Because of this, the Genomic Encyclopedia of Bacteria and Archaea, or GEBA project, was started. The goal of this project was to study other organisms in hopes of finding new genes and processes researchers have not yet stumbled upon. These new discoveries could lead to a number of different breakthroughs in fields such as energy production and pathogenesis, just to name a few. It has been previously concluded that M. ruber is a gram-negative obligate aerobic bacterial species, but since then, very little research has been conducted on it. So that is why I chose to study it for my project. I wanted to gain a better understanding of Myothermus ruber and some of the genes found inside of it. The first thing analyzed in this project was the KEGG pathway maps of both the streptomycin biosynthesis pathway of M. ruber as well as the streptomycin biosynthesis pathway in E. coli. So first thing to note is that in the E. coli streptomycin biosynthesis pathway, the EC number 5422, which is circled in red, represents the locus tag B0688. And the EC number 27724 represents the locus tags B2039 and B3789, which are paralogs of one another. 5422 represents the protein phosphoglucomutase. Phosphoglucomutase converts D-glucose-6-P, otherwise known as D-glucopyranose 6-phosphate, to D-glucose-1-P, also known as alpha-D-glucopyranose 1-phosphate. EC number 27724 represents the protein glucose-1-phosphate thymidyltransferase. This protein converts glucose D1P to DTDP glucose, otherwise known as DTDP alpha-D glucose. On the Myothermus ruber biosynthesis pathway, it can be noted that the same EC numbers are in the same positions as that of the E. coli. EC number 5422 represents the locus tag MRUB2052, and the EC number 27724 represents the locus tags MRUB0628 and MRUB2034. The fact that these KEGG pathway maps show these EC values in the same exact places on these maps is the first suggestion that the Myothermus ruber genes and the E. coli genes are orthologous to one another. Based on the fact that MRUB2052 and B0688 were represented by the same EC number, which was 5422, they were chosen to be analyzed together to determine if they were orthologous to one another. The first thing done was a protein blast, which is a bioinformatics tool, was conducted between MRUB2052 and B0688. As can be seen, 63% of the amino acids found in the proteins were identical to one another. And It can also be seen that there was a very large bit score of 698. What this large bit score means is that the sequences are very identical to one another. There's also a small e-value represented by 0, 0. If an e-value is smaller than 10 times 10 to the negative 100th, it sometimes is given by the symbol 0.0. So that means our e-value is very small. This is good because a very small e-value suggests that this sequence alignment and this sequence similarity is not due to chance. Rather, it's due because they are very similar sequences. A number of other different bioinformatics tools were used to help determine if these genes were orthologous to one another. One of them include the CDD database. So what that does is it compares the sequences, the amino acid sequences of both genes to a COG family. Here it can be seen that both genes matched up to the same COG family, which was COG 0033, with very, very small EC numbers again. Also, a number of different cellular location bioinformatics tools were used to help determine the location of the proteins that these genes code for, and all of them, for both MRUB2052 and B06688, concluded that their protein most likely is in the cytoplasm of the cell. Another bioinformatics tool that was used is compared the amino acid sequences of these genes to a TIGR-FAM protein family. They both match up to the same TIGR-FAM protein family, which is TIGR01132. This TIGR family also suggested that these genes code for the protein phosphoglucomutase. And again, it can be seen that there's very high bit scores and low e-values. And then another bioinformatics tool was PFAM, also compared the amino acid sequences to protein families. Both genes, again, were identical in their matchups with these protein families. All of these bioinformatics tools suggested that both genes are orthologous and that they code for the protein phosphoglucomutase. For MRUB0628 and B2039, which were coded for, or which were represented by the EC number 27724, they also were analyzed using bioinformatics tools. Again, it could be seen that for like the CDD database, they matched up with the same COG family, which was COG 1209. This suggested that they coded for the protein glucose-1-phosphate thymidyltransferase, which was similar, or which is identical to what the KEGG pathway map suggested. It can be seen that they matched up with very, very small e-values. Again, the bioinformatics tools that suggest where the proteins are, or the cellular location, that all suggested that these proteins could be found in the cytoplasm of the cell. TIGR-FAM, protein family bioinformatics tool, suggested that they both belong to the TIGR 01208 protein family, suggesting again that they coded for the glucose-1-phosphate thymidyltransferase protein. So yet again, it can be seen that there is a lot of similarity between both MRUBR 0628 and B2039, suggesting that they are orthologous and that they both code for the protein glucose-1-phosphate thymidyltransferase. Just to get a better look at one of the bioinformatics tools used, this is a alignment of the PFAM top hit between both MRUBR and E. coli. So this is PFAM PF00483. So in panel A, that is represented by MRUBR, and panel B is E. coli. So as can be seen, all the light blue color is the matchup between the P family amino acid sequence and the either MRUBR or E. coli amino acid sequence. The red boxes are the similarity between the M. ruber, and E. coli. So as you can see, there is a lot of similarity between both the protein family as well as the M. ruber and E. coli amino acid sequences. So again, just to recap, all these bioinformatics tools pointed to the fact that MRUB 0628 and B2039 are orthologous to one another and code for the protein glucose 1-phosphate thymidyltransferase. Finally, MRUB 2034 and B3789 were also analyzed through bioinformatics tools. Again, for the protein blast, very high score, very low E-value, suggesting that they had very similar amino acid sequences. They both related to the same COG number, which again was COG 1209, suggesting that they coded for the protein glucose 1-phosphate thymidyltransferase. This makes sense because if we go back to the KEGG pathway map, according to the E. coli streptomycin biosynthesis pathway, 27724 was a paralog, and so is that found in M. ruber. So just to go back to that table, again, the TigerFam protein family, they match to the same protein family with high bit scores and low E-values, and they had the same EC numbers and things like that, all suggesting that these genes were orthologous and coded for the same protein, which is glucose 1-phosphate thymidyltransferase. Just to get a better look of that BLAST sequence I had mentioned before, here you can see that 39% of the amino acids were identical between MRUB 2034 and E. coli B3789. And their bit score is very high with 160 and very low with the value of 2e to the negative 51st. So in conclusion, M. ruber, MRUB 2052, and E. coli B0688 are orthologous to one another and code for the protein phosphoglucomutase. MRUB 0628 and E. coli B2039 are orthologous to one another and code for protein glucose 1-phosphate thymidyltransferase. And MRUB 2034 and E. coli B3789 also code for the protein glucose 1-phosphate thymidyltransferase and are orthologous to one another. After these conclusions were made, a hypothetical site-directed mutagenesis was designed using the NE base changer bioinformatics tool. It was chosen, or MRUB 2052 was chosen to be used for this site-directed mutagenesis because of its high identity to B0688. The histidine at position 60 was chosen to be the amino acid that was to be changed. This is because histidine is known to be very important to protein function. So using any base changer bioinformatics tools, primers were designed to switch histidine or substitute histidine with an alanine, which would most likely result in a loss of protein function.